Good morning, students. So let's begin with the second part of the lesson number 2, that is structure of chromosome, cell cycle and cell division. In earlier part we have studied about the chromosome. Now in this part we are going to discuss number 1, that is cell cycle. What are the different phases of cell cycle? Concept of mitosis, that their phases, that is karyokinesis, its subphases, cytokinesis, importance of mitosis, that is significance, introduction to the meiosis, that is the only thing we are having as a scope of our syllabus, and what is crossing over. Okay, so let's begin with the first point. Cell cycle is defined as a series of events that take place in a cell leading to the duplication of its DNA and the division of cytoplasm. What does this mean? Now if you see, in this definition, these are the two important words: the duplication of DNA and the division of cytoplasm. Means when the cell divides, the first thing what is necessary is the duplication of its DNA. Since it is located inside the nucleus control each and every small activity of the cell. We need to create another controlling center for new cell. Now once the new controlling center, that is nucleus, and the DNA is formed, then what comes is the division of cytoplasm. Fine. Now in this diagram, if you see, the cell cycle is broadly classified into two major phases. This is the one actual called as dividing phase, which is also called as mitosis or M phase, and the one which starts from G1, S and G2. These three phases, these phases are called as interphase. Okay, this G1 is coming after the mitosis phase, isn't it? So therefore G1 is also called as post-mitotic phase, and G1 is the phase which comes prior to the mitosis or M phase. We call this is as pre-mitotic phase. Okay, now let's see all these phases in detail. I have classified the same phases with the help of this flow chart. That cell cycle is divided into interphase and M phase, where M stands for either mitosis or meiosis. We do not have meiosis in detail, so we will focus only on mitosis. G1 phase, S phase and G2 phase, these are the subphases of interphase, while mitosis is broadly classified into karyokinesis and cytokinesis. Karyokinesis will further have four phases: prophase, metaphase, anaphase and telophase. What will happen in interphase? The first G1 phase is also called as gap 1 or growth 1 phase. As you can see on the screen, it is the longest phase of cell cycle. The cell undergo majority of a growth, and the most important event that will occur in the cell are the synthesis of RNA and proteins, division of mitochondria in case of plants and animals, and division of plastids in case of plants only. Correct? Along with this, the most important part which occur in G1 phase is the volume of the cytoplasm doubles, as almost all type of the cell organelles, they are going to double in their numbers. Once this complete, cell enters into the second phase, that is synthesis phase. Now in S phase, more of the DNA is synthesized, chromosomes duplicate, but they remain attached to the centromere. Correct? And the centromere also provides the attachment site of kinetochore. This word is new to you. Correct? So kinetochore is a type of spindle fiber. What is that? We are going to discuss this, but for the time being, please remember only these 3 points. If I want to summarize the S phase in a simple sense, you can write what is happening in S phase is the duplication or replication of DNA and condensation of chromosome. Complete once the cell enters into G2 phase, that is growth 2 phase, where the chromosomes will further condense and the assembly which is required for the initiation of cell division, that is centriole, it also divides and cell is ready for actual division. But why do cell want to divide? The cell divides because of these 4 reasons. Number 1, reproduction. Okay, this is also take place through the activity of dividing cells. For example, amoeba and bacteria, they just divide to reproduce into 2 similar independent cells. But in case of higher animals, that is humans or the plants, there are special cells in the reproductive organs which undergo special kind of cell division. We called it as meiosis, so as to produce the gametes, that is sperm and the ovum. Correct? In case of plant, instead of the sperm, the pollen grains are formed, and instead of ovum, the ovule is formed. For growth, yes, every organism, be it is a plant or animal, will start their life from a single cell. We called it as zygote, and this cell divides repeatedly so as to form the clusters of cells. We call them as tissues. Tissues will form organ will form an organism. Correct? Third reason is for repair. Apart from the normal wear and tear of the tissues in the body, there might be an accidental injuries. One may develop a cut, correct, or can develop the fracture. So repair of such injuries is again through take place by cell division, and it will cover up the gap. A replacement, I would like to give an example of RBCs whose lifespan is 120 days, right? So if I want to convert it into the time analogy, every second we are losing 2 million RBCs from our body. So they need to be replaced. So from where they will be replaced? The cell division that will take place in red bone marrow will replace them. Right? We'll go ahead. Mitosis, this is what is the definition: an equational division in which one parent cell divides to form the 2 daughter cells. In mitosis, the number of chromosomes will remain same at each stage. It occurs only in eukaryotic cells, that is in case of human and plants, but that too in case of human, it will not take place in a neuron. Apart from neuron, all body cells will divide mitotically. Okay, so if anybody asks you what is the location of mitosis, your answer will be all body cell except neuron and reproductive organs. Neuron, because of absence of centriole, will not divide, and as I told you, reproductive organs, they will divide by meiosis, they will not divide by mitosis. Right? The mitosis is completed into 2 phases. Number 1 is called as karyokinesis, that is division of nucleus. Number 2 is cytokinesis, that is division of cytoplasm. We'll see these 2 phases in detail one by one. So this is the first phase of karyokinesis, that is prophase. Now here I am going to take the examples of animal cell only, but it doesn't mean that it will not occur in the plant cell. Correct? What I want, simple, you to please refer the page number 14 of our textbook, where all the stages of mitosis and mitosis in plant cell and animal cell is summarized. You have to draw and practice all the diagrams from that page. Okay. Now what are the changes that will take place in prophase? Correct. Once the cell completes its interphase, cell enter into the prophase. The nuclear membrane and the nucleolus will disappear. Chromosome will start appearing and clearly visible. Centriole will start moving apart from each other, and the spindle fiber starts appearing. One by one we can see in this diagram. If you see here, this is what is the nucleus and this is what is the cell. Now this nuclear membrane will start disappearing. It will break into the number of pieces. Chromatin material, now it has started condensing into chromosome, forming centromere and two chromatids. Nucleolus, if you can see, it is going to be invisible, or it will disintegrate, and then the nucleoplasm will mix with ectoplasm because there will be no membrane. Apart from that, if you see, this is these two bar bodies, they are called as centriole. They are also divided. They have started forming the spindle fibers, right? And the spindle fibers, along that, they have started moving apart from each other. Once they start moving apart from each other, they will take the pole position. See, pole means extremities of an axis, and equator means the middle plane which divides the cell. Correct? So take these two concepts in that context. Don't consider pole means exactly they will go far away from each other. Just they will be exactly opposite to each other. Spindle fibers originate from the centrioles, and they are of two types. As I told you, what are spindle fibers? They are the mitotic spindle formed from microtubules in case of plant and centrioles in case of animal cell. Remember, centrioles are absent in plant cell. So how does it possible that plant cell will divide? So the plant cell divide by using microtubules. There are two types of spindle fibers. Number one is polar fiber. Second is kinetochore fibers. Polar fibers, they will extend from one pole of the cell to the opposite pole. To simplify this, polar fibers, they are extended from one centriole to the another centriole, because we just discussed that centriole will take pole position. While kinetochore fibers, they will extend from a pole, that is centriole, to the centromere of chromosome. Now, once these centrioles or the pole, they start forming the spindle fiber, they appear like an asterisk, which you can see in your mobile when you type star hash some number. So that time that star is not a star, that is an asterisk. So therefore we can say that in animal cells the asters are formed, and these asters are the short fibers which are radiating from the centriole. Student, to understand 1, 2 and these 3 concepts, I want you to observe the next diagram. In this diagram you can see these are the two poles, that is, this is one centriole, this is another centriole. They have started forming the rays or the spindle fiber, hence we are calling it as asters. Now carefully observe that both these centrioles, they are forming two different fibers, one which are shown in the black line and another with the blue color. This blue color line first we will discuss. They are originating from one centriole going to the another centriole, that is from one pole to another pole. We call these blue color lines as polar fibers. We call them as what? Polar fibers. Second, this black color line originating from one centriole attaching to the centromere of the chromosome. Number 1, number 2, number 3 and number 4. Remember students, we are having 46 chromosomes in our cells, but it is not possible to show always that many number of chromosomes. So mostly in your exam also, diagram based question if they ask, then that time either they will show you the 4 chromosomes or they will show you the 6 chromosomes, right? We'll go ahead. Once the prophase is complete, cell will enter into metaphase. In metaphase, all the chromosomes attached to the kinetochore fibers line up at the equator. What is equator? The middle line, correct? And the metaphase is over. Cell enters into, that is, anaphase. In anaphase, it is a very short phase and it occurs so rapidly that sister chromatids will pull apart to the opposite poles of the cell by using kinetochore fibers. Opposite pole means nothing but the centrioles. All these fibers which are attached to the chromosome, that is by centromere, they will be contracted, they will be pulled. Once they are pulled, the chromosome will separate and they will slowly start migrating towards the centriole. These 4 will migrate towards this side centriole, while these 4 will go to the opposite centriole. Once this is over, the cell will end to telophase. Telo stand for end and phase stand for state. What will happen in this stage? The daughter chromosome will finally reach to the opposite pole. As compared to the earlier slide, if you see here, now here chromosome is almost converted into chromatids. As spindle fibers are disappeared, nuclear membrane nucleolus will reappear and there is a cleavage formation starts in animal cell. Remember, in our book, the cleavage formation has shown in the anaphase, which is a mistake. Okay, always remember cleavage formation in animal cell starts in telophase only. Okay, we'll go ahead. Once this is completed, cell will enter into cytokinesis. Cytokinesis means it is a division of cytoplasm. The division of cell into 2 identical halves, that is called as daughter cells, and this is done in case of plant cell by the formation of cell plate, and this cell plate formation is done by using Golgi vesicles. Golgi vesicles, they are the small vesicles which are produced from Golgi apparatus. But in case of animal cell, the cleavage furrow, it will form further and it will split the cell into 2. To understand these 2 points, look at this diagram. As you can see in this diagram, the furrow which was started in the telophase, finally from left side it will come and reach to the central point, and from right side it also come here and meet at the center. Once the furrows or the constriction from both the sides, they will meet at the center, the cell will get separate. Okay, but in case of plant cell, as I told you, at the equator there is a lining up of the Golgi vesicles. You can see these small round shaped structures. These are called as Golgi vesicles. All these Golgi vesicles, they arranged in a line. The wall in between them will dissolve and whatever amount of cellulose is present in that Golgi vesicle, it will be planted over here. Planted, I mean to say, they will form the partition. And once this is formed, this partition is called as cell plate, responsible for the division of cell into 2 daughter cells. With this, the mitosis is over. Right? Now, why this mitosis is necessary, and why, or simply what is the significance of this mitosis? We already have discussed this, correct? So, mitosis is required for the growth or to increase in body size due to the formation of new cells. repair of the damaged and wounded tissues, replacement of old and dead cells. Asexual reproduction in case of unicellular organism will take place with the help of mitosis, for example amoeba or yeast cell, and this is also responsible or required to maintain the same number of chromosomes. Students, how much time this cell division or cell cycle continues? You have to remember that some places the cell cycle will stop permanently, some places it will stop temporarily, and at other cells till it is needed it will continue. Okay, for example, in case of brain cells, once they are formed in the embryo they do not divide, because I told you that neuron do not have the centriole. In case of liver cell, they divide only once every year. Surface skin cells, since they are replaced because the outermost layer is made up of cornified layers, then there is a continuous mitosis will take place. In case of plant, the cells at the growing points, that is meristematic tissues, will divide rapidly. Correct? Specialized germinal cells, that is in case of ovary and testis. Correct? In animals, and ovary and anthers in case of plants, they will undergo meiosis so as to produce sex cells only after they become functional, that is once they attain the puberty. But sometimes this cell division may go uncontrolled manner, and that result into the formation of tumor. But remember that tumor may or may not be cancerous. Coming back to this mitosis part, this is what is the difference between mitosis in animal cell and mitosis in plant cell. In case of animal cell, there is a formation of asters. In case of plant cell, there is no formation of asters. Cytokinesis completed by furrow formation, and here by the cell plate formation. Animal cell, the mitosis will occur throughout the body parts, but in plant cells it occurs only at the growing tips, that is meristematic tissues. Questions are likely to be asked, and I want at least you to remember the first 2 points from this difference between. Okay. The second type of division is meiosis. It will take place in reproductive organs. In case of animal, that is testis and ovary, and in case of plant, anthers and ovary. The most important feature of this meiosis, that is the number of chromosomes becomes half. Now here I would like to explain you 2 important terms which I want you to note down. You can pause this video and then note down. Number 1 is diploid and haploid. What do you mean by the word diploid? Diploid number of chromosomes, which means the full number of chromosome. Correct? And haploid number means, or haploid chromosome means half of that number. The diploid number of chromosome is represented by 2n, and haploid number is represented by n. Now in case of human, 2n is equal to 46 and therefore n is equal to 23. You can do this simple maths calculation. So what does this mean? My skin cells, they are having 46 number of chromosomes, that is 2n, that is diploids. But the sperms which are produced, they will not have 46 chromosomes, they are having 23 chromosomes. So therefore, when 23 chromosome present in the sperm fuses with the 23 chromosome present in ovum, once the zygote form, again they will have 23 plus 23, that is 46 number of chromosome, which means haploid converts into diploid. Don't forget these 2 concepts, students, diploid and haploid. With respect to this, we are also having diploid cells, means the cells which are having full number of chromosome, and haploid cells, which means the cells is having half number of chromosome. So if I ask you, is the liver diploid cell? Your answer is yes. And is ovum haploid? Your answer is also yes, because of the explanation what I just gave you. We'll go ahead. It is called as reduction division because the number of chromosomes becoming half. it is completed into two steps, meiosis 1 and meiosis 2. In meiosis 1, the chromosome numbers will reduce to the half, and in meiosis 2, the reduced number of chromosome will remain same, and at the end of meiosis, always remember there are the formation of 4 cells. Okay, I'll show you this meiosis step with the help of diagram, but remember this is just for your information. We'll go ahead. Now as you can see, the meiosis is divided into two parts, meiosis 1 and meiosis 2. In meiosis 1, there is a segregation of homologous pair that are separated, reducing the chromosome numbers by half. So if you see here, once the interphase is completed, which will take place basically in the germ cells in the gonads. Gonads means primary reproductive organs. Meiosis is completed. So at meiosis 1, once it completes, it will divide into two daughter cells. Once it will divide into two daughter cells, the number of chromosomes are half. Means if here there are 46, then here it will become 23. Once this is happen, both these two cells, they will enter into meiosis 2, and this cell continues to form two daughter cell with 23 and 23 chromosome number. This another second cell which was formed at the end of meiosis 1 further undergoes division and also form two cells with 23 and 23 number of chromosome. If you refer the diagram from page number 23 of our textbook, the same thing you can see. Here only I have not mentioned the diploid and haploid number, but please remember the cell here is having 2n number of chromosome. These two will have nn number of chromosome, and these four are also having nn. Correct? Now if you see here, this second division, meiosis 2, is also called as mitosis. A question can be asked to all of you, give scientific reason. Why meiosis 2 is called as mitosis? Because once the cell complete meiosis 1, entering into the meiosis 2, they are having 23 chromosomes in each. Correct? The cell is having 23 chromosomes. This is also going to divide, but when they divide, the chromosome numbers are remain same, and if the chromosome number remains same, we called it as mitosis, right? So only meiosis 2 is called as mitosis. Meiosis 1 is not called as mitosis, but together is the part of meiosis. Therefore we say one cell will divide into 4 daughter cells at the end of meiosis, right? Significance of meiosis: chromosome numbers become half, mixing of the genes is possible, hence they are creating variation, and this variation is occurred by using the technique called as crossing over. Now what is crossing over? I'll explain with the help of diagram. These two are called as homologous chromosome. What is the meaning of the word homologous chromosome? The chromosomes which are having same shape and the size receive from one parent each, called as homologous chromosome. Pause this video and note down this definition. Listen it again. So this homologous chromosome are having chromatids. The non-sister chromatids, which means chromatids from each chromosome, will overlap, as you can see in this diagram. The blue color chromatid is overlap with the red one. So this overlapping is called as crossing over, and the point where the crossing over takes place is called as chiasma. So this chiasma, once it is formed, the crossing over completes. These two chromatids separates with each other, but once they get separate, you can see here the genetic material is exchanged, and because of this exchange of genetic material, we are having variation among the organisms. Okay, this is the major difference between mitosis and meiosis. The number of chromosomes remains same in mitosis, but in meiosis it will become half. Mitosis occurs in the somatic body cells, while meiosis occurs in the reproductive cell. Mitosis continues throughout the life of an organism, but meiosis continues only till the organism is active reproductively. Nucleus in case of mitosis divides only one time. In case of meiosis, it will divides two times. As you can see, meiosis 1 and meiosis 2. Mitosis, it occurs for growth, repair and replacement, while meiosis, it will occur only for the gamete formation. The point which I have not mentioned over here, I want you to remember or note it down somewhere. At the end of mitosis, 2 daughter cells are produced, and at the end of meiosis, 4 daughter cells are produced. Okay? With this, thank you so much for watching this video. I will request you to please read the lesson thoroughly and carefully. Try to solve the progress check question and also your textual questions, those who are the objective one. Please practice and remember all the stages of mitosis in case of animal cell and in case of plant cell. Thank you so much.